So, Assalamu alaikum. My name is Saad Meliti, a third year medical student, and today, inshallah, I will be going over clinical microbiology, and specifically, I'm going to be going over the other stuff, okay? So, no more bacteria. We're now covering the protozoa, the helminths, the what's it called? The fungi. I won't be covering viruses though, unfortunately. But hopefully this will be enough for all of Muhabbat's lectures specifically, okay? So let's get into it. This is my email. I don't know if anyone's emailing me. I don't check that. Here's my number. If you guys want to message me, generally, if you guys want to have, or if you guys have any questions you guys want to ask me, I'll be available to answer you guys as soon as possible. But generally, clinical microbiology is it's very memorization heavy, so that's why hopefully this session is going to be a quick review. I want to quickly review over everything. Um, and inshallah it'll be a nice review for you guys. Generally, this stuff is kind of just memorization. Hopefully there won't be much questions, but if you do have a question, just let me know. So let's get into it. Parasites. I hate this pen. Parasites. Okay. Now parasites. When we want to talk about parasites, we kind of have to understand what a parasite is. So I won't get into too much details about all of this, but as we probably know from high school, mutualism is when two organisms benefit from each other. Commensalism is when one what's it called organism benefits from the other, and the other is left unaffected. And parasitism is when one organism benefits from the other, like the tick feeding on the cat's blood, and the cat actively gets harmed in this process. Okay, so the cat gets skin irritation, or even much worse. Okay, so that's what parasitism is. Okay, so there are a few definitions that Muhammad starts her lecture off with, and we have to go through all of them, and you guys should know all of them for hopefully the exam, yani. So first off, pretty obvious ones, ectoparasite. Okay, so it's a parasite that lives on the host outside. Okay, for example, lice. So you guys see the lice here. It's on the patient's head. Okay, and it's living outside, but it's still causing the patient harm. Okay. Next off, we have endoparasites, and these live inside the host. So this is, a worm. We're gonna talk about worms later on, and these kind of live inside of the gut. Okay, and these, feed on your mucosa or whatever, and these are inside the cell. Okay, these are endoparasites. Okay. Now, when we talk about parasites, we have parasites that need a host to survive. Okay, so they need a host. This is known as an obligate parasite. Okay, so it has to have a host to survive. On the other hand, facultative parasites can live freely or as a parasite. So it can live outside a human body, can live inside a human body. It doesn't make a difference. Now, accidental parasites, these are parasites that infect an unusual host. So we're going to talk later about Echinococcus, I think. I forgot. But it's a worm that's usually found in dogs, okay? But sometimes there's a chance that this worm infects humans, okay? Instead of going through the normal life cycle of, the dog and the sheep, the dog and the sheep, it eventually ends up in a human by accident, okay? That's what an accidental parasite is, okay? These usually shouldn't infect us. Now, a host is the organism harmed by the parasite. So in the example I talked about earlier, the bee, not the bee, sorry, the tick feeding off of the cat. The cat is the host in this case because the cat's the one getting harmed. Okay, and there are two types of hosts. We have the definitive host, where sexual reproduction happens. Okay, and this is specifically where the adult parasite is. Okay, so the adult parasite is found in the definitive host. The intermediate host is where asexual reproduction happens, and this is where we see the larval stage. Okay, larva, baby form of the parasite. This is in the intermediate host. Okay, and now there are two types, or there are multiple types of hosts. Sorry, there's the transport host, and it carries the parasite without growth or multiplication. This is very important because it separates it from the reservoir host, which stores the parasite viably. Okay, so the reservoir host also carries the parasite, but they don't have symptoms. There is growth and multiplication. Okay, that differentiates it from transport host. Okay, now the accidental host, it goes back to what I talked about earlier, the accidental parasite. The accidental host is just the wrong host for the parasite. So in the dog tapeworm, for example, humans are the accidental host. Okay. Now, a life cycle is the sequence of stages a parasite goes through from one generation of adults to the next. So, parasites, we're going to talk about each life cycle. They usually follow a life cycle. They infect a human, get shed out, infect another human. They all have different life cycles that we're going to talk about. And there's two different types of life cycles. We have the indirect life cycle, which needs at least one intermediate host. And an intermediate host, like we said, is where asexual reproduction happens. So in malaria, for example, the Anopheles mosquito is the intermediate host. So we call this an indirect life cycle. Direct life cycles, on the other hand, pass from one host to another of the same species with no intermediate host. Okay, no intermediate host is important. So, for example, a direct life cycle would look like this. It goes from human back to human back to human. There's no other species involved in this. Okay. Now, when we talk about parasites, we can divide it into protozoa or metazoa. Now, simply, protozoa are just single-celled organisms. Okay, like this one, for example. Multicellular organisms are metazoa, and they can then further be divided into the worms, which we're going to talk about later. Okay. For now, we're going to be covering the protozoa. Okay. So let's get into protozoa. We first have our first protozoa, which is Giardia intestinalis. Okay. Giardia intestinalis is a flagellate protozoa. So it has flagella. Okay. Like this, like this, like this one. Okay. And the trophozoite form. So there's two forms of Giardia. Giardia has the trophozoite form and the cyst form. Okay, the trophozoite form has what's it called? Where's my pen? Two nuclei as such, and four flagella. Okay, four, one, two, three, four. Okay, the cyst on the other hand has four nuclei, and gives rise to two trophozoites. Now the picture here shows two nuclei, but I'm gonna show you pictures later. I think in other pictures, Yani. It actually has four nuclei, and then it gives rise to two trophozoites. Okay, and the trophozoites are the one with two nuclei. Okay, now specifically, some things you need to know about Giardia is that an IgA deficiency can predispose to symptomatic infection. So IgA protects the mucosal lining of your stomach, which Giardia infects. So if you have an IgA deficiency, you might be predisposed to symptomatic infection of Giardia. And when we want to get rid of the cysts of Giardia, chlorination doesn't work. So to get rid of the cysts, filtration works. Okay, filtration is important in getting rid of the Giardia cysts. Okay. And last but not least, mammals and humans act as a reservoir asymptomatically. So this doesn't only infect humans, it can also infect mammals. Okay, and both of us can act as reservoirs. Okay. Now, in the life cycle, transmission can start by the ingestion of cysts. So in this case, in this life cycle, the infective stage that infects humans is the cysts, like right here. Okay, the cyst has once again four nuclei. You can count them like this, Yani. Okay. Uh, Now, the cysts are going to be in fecally contaminated food or water. So, the cysts are released in feces, can contaminate food and water, which then can get ingested. Okay. And then the cysts turn into trophozoites that attach to the gut wall. But when they attach to the gut wall, they do not invade the mucosa. Okay. They don't invade the mucosa and they don't enter the bloodstream. Okay. This will be important when we, what's it called? Differentiate it with entamoeba, I think. So, just by attaching to the gut wall, trophozoites can still cause inflammation of the gut wall, and this leads to malabsorption of proteins and fats. Okay, so you can have different kind of symptoms, although not too important: weight loss, diarrhea, stomach cramps, flatulence. Um, a lot of things you can think of when it comes to malabsorption of proteins and fats. Okay, so when we want to diagnose Giardia lamblia, like I said, the cysts and even the trophozoites are found in the stool. Okay, and specifically the diagnostic stage of Giardia lamblia is the trophozoite. Now, the trophozoite is very characteristic. Like it looks very, special. Okay, and every kind of parasite has its own special, form that can be diagnosed by some kind of microscopy. And the trophozoite looks like this. If you see it in the stool, you know that the patient's most likely infected by Giardia lamblia. Okay, and obviously you can also see the cysts in the stool as well. Other methods of detection include ELISA and PCR, but this isn't too important. The gold standard is obviously seeing the trophozoite in what's it called in stool samples. Okay. Next up, we have Entamoeba histolytica. It's an amoebic protozoa, so it's an amoebia or whatever it's called, and it's amoeba. Sorry. And amoebas don't have flagella; they move with their cytoplasm, right? Now, there's two forms once again for this. There's the what's it called? The trophozoite and the cyst. Now, the trophozoite, it's important to know that we can only find the trophozoite in diarrheal stool. Okay, so watery stool is going to have the trophozoite. The cyst, on the other hand, is found in normal non-diarrheal stool. Okay, so it's important to know that. Okay. Now, the cyst of Entamoeba histolytica has four nuclei, and keep this in mind. It's like Giardia lamblia. Keep this in mind for diagnosis later. Okay, and the trophozoite has a single nucleus with an evening lining of chromatin around the nucleus. And it has it has a single central nucleus like this. So this is the nucleus, right? And there's an even lining of chromatin around it. So this circle right here is chromatin. You can see how it's evenly lined. And then there's a prominent central nucleolus. This is the nucleolus. It's in a central position, if you can tell. Now, in terms of the life cycle, the transmission starts by ingestion of cysts by the fecal oral route. Okay. Now, just like Giardia lamblia, it's the same thing. The infective stage is the cyst. So someone who ingests the cysts, contaminated stuff that's by the fecal oral route can get infected by Entamoeba histolytica. And I don't know why the doctor is obsessed with this, but she also wants you guys to know, even for Giardia lamblia, since both of these are through the fecal oral route, it's very possible for homosexuals to, transmit the disease through anal oral transmission. Okay, she might ask that. I don't know why she wants you guys to know that, but I guess it's high yield. Okay. The thing that differentiates this from Giardia to some degree is that there's no animal reservoir. Okay. We said that, what's it called? Giardia has animal and human reservoirs. Entamoeba is only found in humans. Okay. So humans infect humans. Okay. So the infective stage is the cyst. We can diagnose the cysts and the trophozoites in the stool. Okay. Now, when it reaches the gut, when the trophozoites invade the colon, okay, they cause necrosis of the epithelium, and they can invade the blood, systematically affect the liver, cause liver abscess. Okay. And something very characteristic of Entamoeba histolytica is this picture right here. It's a flask-shaped ulcer. Okay. It looks like a bit of a flask inside the gut mucosa. And this is very characteristic of Entamoeba histolytica. So keep on the lookout for this because unlike Giardia, Entamoeba actually invades the epithelium and causes necrosis. Okay? Now, there are different types of Entamoeba. How do we differentiate, Entamoeba histolytica specifically, the disease-causing Entamoeba from other different amoebas? Well, first off, the nucleus, okay? The nucleus, like we said, we can see fine, going around in a circle, very fine chromatin granules, okay, going around. It looks very smooth, okay? Unlike, you know, um, over here, okay? This is Entamoeba coli, for example. It's another type of Entamoeba. The chromatin granules are just very, weird. Like one of them is bigger than the other. It doesn't seem like it's following a proper order. So that's what we mean by fine chromatin granules. Another thing is that Entamoeba histolytica has a central nucleus, or sorry, nucleolus, okay? And it's kind of small central, okay? Now, Entamoeba coli, on the other hand, look at it. It's not centered. It's off center. It's huge. It doesn't look, fine at all. And you're gonna see this in a lot of other different types of entamoeba. But entamoeba histolytica specifically, has this kind of very fine, very, organized kind of nucleus. Okay. Another thing is that entamoeba histolytica, like we said, has four. What's it called? Has four nuclei in its cyst. Okay, and it's a bit smaller. The cysts are a bit smaller than other cysts. Okay. But for example, this is Entamoeba coli. It has like what, one, two, three, four, five, six, seven, nuclei. Um, but Entamoeba histolytica has four. Okay, that's diagnostic for Entamoeba histolytica. Okay. Now the Entamoeba histolytica trophozoite has directional movement as well. So if we went back to Entamoeba coli, for example, we'd look at its movement and we'd realize that the movement isn't very directional. It doesn't have a purpose. Entamoeba histolytica, on the other hand, every kind of movement, is directional. It has a purpose. It's going somewhere. Okay, that's something to differentiate it from other organisms. I think that's it for Entamoeba histolytica. Now, I kind of didn't add these in my slides, at the beginning because she never really asks about prevention. Prevention is very low yield for her lectures, and they're very common sense. But I heard that she emphasized on it in her review session, so I added it. So to start with, when you guys think of fecal oral transmission, what do you think of? um, Washing your hands probably, cleaning the water, all that kind of stuff. But all of that is seen in the pictures. You just want to make sure the food and the water you're drinking isn't contaminated. You want to make sure you're washing your hands to not spread the disease, all that kind of stuff to prevent, all of these diseases. Next, moving on, we have the Plasmodium species. Okay, this is another parasite, and these cause malaria. Okay, And in the Plasmodium species, there's a sexual cycle and an asexual cycle. So the sexual cycle is called sporogeny because sporozoites are produced in this cycle. The asexual cycle is called schizogeny because schisms are made. Okay? I hope that makes sense. And the vector or the definitive host of Plasmodium species is the female Anopheles mosquito. Okay, the female Anopheles mosquito is what infects humans with malaria, this disease. Okay, so saliva from a mosquito bite can enter the human and give the human sporozoites. Okay, so the mosquito has a bunch of sporozoites in their body, and it spreads it to the human, and they go around the blood until they end up in the liver and they get taken up by hepatocytes. Okay, now this is called the exoerythrocytic cycle. The exoerythrocytic cycle is in the hepatocytes, and it's called exoerythrocytic because it's outside of the blood cells. Okay, so in the liver cell, it forms something called a schizont, and the schizont ruptures and releases a bunch of merozoites. Okay, these merozoites then go on to infect the blood cells. Okay, now what's interesting is that there are different types of Plasmodium, P. vivax and P. ovale, and these produce a latent form in the liver cells called a hypnozoite. Now, this hypnozoite is latent in the liver cells, it's chilling in the liver cells, and it can randomly reactivate at some point and cause relapse. Okay, I hope that makes sense. Um, so once the merozoites are released from the liver cells, released from the schizonts, they infect red blood cells, and this starts the erythrocytic phase. Okay, once the erythrocytic phase starts, they become trophozoites. Okay, so over here you can see the merozoites infecting red blood cells. It goes to the red blood cell, and it becomes a trophozoite. This is called the ring stage. Okay, in the red blood cell, and this is the once again the erythrocytic phase. Okay, now these trophozoites follow, its own cycle, okay, making a schizont again, rupturing, reinfecting more blood cells. Some of them can go on to form gametocytes, okay? This is what a gametocyte looks like, and they kind of look like banana shaped on blood smears. We're going to see it in a bit. These gametocytes, can get, ingested by the mosquito. The mosquito takes the gametocytes, and then let's talk about it right now. The zygote becomes a motile eukaryote. Okay, this is the eukaryote, and then it goes to the gut wall of the mosquito, becomes an oocyst over here, and this oocyst releases a bunch of sporozoites. Okay, this is sporogeny. Okay, now these sporozoites inside the mosquito can then go on to infect another human being. Okay, so something to know about malaria again is that, or specifically Plasmodium. There's a species of malaria called Plasmodium malaria. Okay, and this is the only parasite with a quartan cycle. The rest have a tertian cycle, so it takes 48 hours. Okay, Plasmodium malaria takes 72 hours for its cycle. Okay, I hope that makes sense. Now, when we want to diagnose Plasmodium, we can diagnose with a thick or thin blood smear. Okay, and the diagnosis is through the identification of trophozoites or gametocytes, whatever you want. So we can see here a thin smear. We can see the trophozoites inside the red blood cells. They're all here. They're chilling. If you go over here, this is another what's it called? Thin smear. You can see the gametocytes, and this is a thick smear. This is what a thick smear looks like. You can't really see the red blood cells, but you can still see the trophozoites here. They're very visible. So you can see it on both thick and thin smears, and you're looking for the trophozoites once again or the gametocytes of Plasmodium. Now, we can also rapidly detect Plasmodium using, molecular biology. First off, we have the enzyme PDLH, lactate dehydrogenase, and it's for all species of Plasmodium. Okay, this is Plasmodium lactate dehydrogenase. This is for all species of Plasmodium. Um, it detects every species. And then there's histidine-rich protein 2, TFHRP2. It's specifically for Plasmodium falciparum, okay? So Plasmodium falciparum has this specific histidine-rich protein 2, which we can detect. So for example here, we have the anti-falciparum antibody, which is the histidine-rich protein. We have the anti-malaria antibody and the control antibody. And using this, we can, you guys don't really need to know how to interpret all of this, but we can use all of this to kind of, what's it called? What was I going to say? Diagnose malaria. Either figure out if it's, a random kind of malaria or find out if it's falciparum, okay? So now let's get into Leishmania species. So there's three different types of Leishmania, or specifically three different disease manifestations. We have visceral leishmaniasis caused by L. Donovani. Okay, L. donovani causes visceral leishmaniasis. L. tropica, L. mexicana causes cutaneous leishmaniasis, and Leishmania brasiliensis causes mucocutaneous leishmaniasis. Okay, so I'm gonna try and repeat these over and over again because you kind of just do have to memorize the names. So Donovani once again for visceral, Tropica and Mexicana for cutaneous, and Brasiliensis for mucocutaneous. Okay. Now, in this kind of what's it called life cycle, we have the vector, which is the female sandfly. Okay, so it's the sandfly in this case, and the sandfly bites a human, introduces promastigotes into the host. Okay, so the promastigotes enter the host, and they are engulfed by macrophages, where they then transform into amastigotes. So inside the macrophage here, it transforms into an amastigote. Now, the amastigote can then be ingested by the sandfly again. So the female sandfly can go to a human, take up the amastigote, and then the cycle repeats again. Okay. So visceral leishmaniasis, we said, is caused by L. donovani, and it affects the organs of the reticuloendothelial system. So the liver, the spleen, and bone marrow, those are the most severely affected. And you can see a child here. This is just a picture I took on my own from Google or something. This patient has hepatosplenomegaly, so he has both his liver and his spleen, they're enlarged. And this is visceral leishmaniasis. It's very dangerous. Now we have cutaneous leishmaniasis. We said, what's it called? L. mexicana, what was the other one? L. tropica, L. mexicana. These cause cutaneous leishmaniasis, and you can just see, skin manifestations. Okay. Then we have mucocutaneous leishmaniasis, arguably the worst one. There are horrible pictures of mucocutaneous leishmaniasis out there, and this is specifically caused by L. brasiliensis. Okay, and you can see that it's attacking a mucous membrane. Um, that's mucocutaneous. That's what we mean by that. Okay. So in terms of diagnosis, we can visualize the amastigotes under a microscope. So let's say we have cutaneous leishmaniasis. We take a skin sore. We kind of take a biopsy of it. We take a biopsy of the bone marrow. We can see the amastigotes, and they're inside of a macrophage. Okay, this is diagnostic for what's it called, leishmaniasis. This is another picture of the promastigotes. Now, the promastigotes specifically are just the infective stage. So it's not too important to visualize this. This is just a picture for you guys' understanding. So this is the promastigotes. Okay, the infective stage. Next, moving on, we have Trypanosoma cruzi. It's a life cycle that involves the kissing bug, the reduviid bug. Okay, now this disease causes a bunch of stuff like the megacolon, the dilated hearts, arrhythmias, whatever, a bunch of stuff. You guys don't need to know too much about it. Now, this can be transmitted through either blood transfusions, organ transplants, transplacentally, or even through laboratory accidents. Okay, so in this life cycle, this is what the reduviid bug looks like. When the bug bites you, okay, it also drops feces, okay, and these feces can enter the wound, and these feces have trypomastigotes, okay. Now the trypomastigotes enter your blood, become non-flagellated mastigotes that are inside host cells. So, last disease, leishmaniasis, we said it's in macrophages, mastigotes inside macrophages. In Chagas disease or Trypanosoma cruzi, the mastigotes are just inside any random host cell. Okay, and they kind of divide there. The mastigotes in this case can actually leave the macrophage, or sorry, the cell, not macrophage, and become trypomastigotes again. And these re-enter the blood and can be uptaken by the kissing bud once more. Okay. Now, in the bug's gut, it's important to know that these trypomastigotes then become epimastigotes, and the epimastigotes multiply and become trypomastigotes again inside the bug, and the bug will once again infect you. The infective stage is the trypomastigotes. Okay, so in terms of diagnosis, like we said, it can either be inside cells as an amastigote or in the blood as a trypomastigote. So if we take a blood smear, we'll be able to visualize the trypomastigote in a patient, or if we take a biopsy of a patient's tissue, we'll be able to see a mastigote nests inside the biopsy. Okay, any infected organ or whatever, we'll see this nest of amastigotes kind of chilling like that. Okay, so that's it for what's it called, Trypanosoma cruzi. Next, we're moving to sleeping sickness or African trypanosomiasis, and this is caused by two organisms, Trypanosoma brucei gambiense, which causes West African sleeping sickness, and this is important for humans. Trypanosoma brucei rhodiense causes East African sleeping sickness. It's related to animals. It's not too important. And once again, this is a picture of the vector, the fly, the tsetse fly. Now, specifically, this is what a tsetse fly looks like, or the picture I talked about earlier. The tsetse fly injects metacyclic tropomastigotes inside, the skin through saliva, and these multiply and become a trypomastigotes in the blood, and they just multiply. Now, what's special about, sleeping sickness is that the other two diseases we talked about, cruzi, what's it called? Trypanosoma cruzi, and the other one, the leishmaniasis, they both have a mastigote phases. Okay, sleeping sickness, the parasites Trypanosoma brucei does not form a mastigotes, and you're just going to see in the blood trypomastigotes roaming around as such. So there's no kind of phase like that. Okay, a mastigote phase. Okay, so things to know is that these trypomastigotes being in the blood, they can spread to the lymph nodes, they can spread to the brain, and if they spread to the brain, they can cause demyelinating encephalitis. Okay, not too important to know exactly what this is. It's more like neuro stuff, but basically, when it's in the brain, eventually can lead to sleepiness. Sleepiness can progress all the way to coma. This is why we call it the sleeping sickness. Okay, and for diagnosis, we can see once again the trypomastigotes shown on microscopy on a blood smear. But another interesting thing is that because it infects the brain, okay, it's also possible. Uh, Let me continue my arrow. I thought it was down this. Okay, it's also possible to see it in the CSF. Okay, so if we took a cerebrospinal fluid sample, we might be able to see this parasite roaming around in the CSF. Okay, now I don't know about you guys, but my dad used to tell me a lot as a child not to get near stray cats because they'd give me toxoplasmosis. Maybe that's just my dad, but it is a disease, and the definitive host is a cat. Okay, we are not supposed to get infected by this. Okay, now uh, in humans, the way we get this by accident, it's usually acquired by ingestion of cysts. They can either be in uncooked meat or even cysts like that are in cat feces. Okay, we'll talk about that in a second. So, let's say there's animals that are infected by tissue cysts. Okay, so in their meat there are literally cysts, and let's say we don't cook the cysts properly. Okay, a guy can eat the cysts and he can just get infected straight up. Or let's say a cat, takes a dump in their litter box, and maybe you cleaned up the litter, but you didn't wash your hands properly. You didn't, I don't know why you wouldn't have gloves. Maybe you got infected by the cysts, you spread it to your food, and you ingest the food. You can pass it on to yourself. That's also another way of, what's it called? Getting toxoplasmosis. Okay. Okay. So, like I said, wait, hopefully this works. Okay. So after invading the gut and getting eaten by macrophages, so once the cysts infect us humans, they get eaten by macrophages, and then they differentiate into rapidly multiplying tachyzoids. Okay, this word is important, tachyzoids. Now, luckily for us, the parasites, although they are rapidly dividing, they enter the brain, muscle, other tissues. Our immune system kind of slows these down, and they become bradyzoids. So the tachyzoids eventually become bradyzoids and become tissue cysts inside our body. And these are diagnostic. Okay, so if we take a tissue biopsy of us, we can diagnose toxoplasma by looking at these tissue cysts. Okay, and even in immunocompromised patients, these cysts might actually rupture and release more parasites in the body. Okay, now in Toxoplasma gondii, cell-mediated immunity is important, and it plays the major role in limiting the spread of tachyzoids, like I said. And pregnant women can spread this onto their offspring, but they can only pass it on if they are infected during pregnancy. Okay, so if they're infected, before pregnancy, the bradyzoids will form. So what's it called? So basically, the baby has to get infected by the tachyzoids. So if the mother was infected by tachyzoids, before she got pregnant, the tachyzoids would have already become bradyzoids. Does that make sense? Um, so obviously, a mother with bradyzoids won't spread the infection to her children. If that makes sense. Another thing I didn't mention is that it can actually also pass on through blood. So if a patient with tachyzoids in their blood gives a blood transfusion or even an organ transplant to another patient, they can spread this infection to someone else. I think that's it for Toxoplasma gondii. This is just a picture of a tissue cyst. So we can take a histological section of a lesion and find the cyst and diagnose it. But let's say we're looking at a baby. Obviously, we can't just take a biopsy from a baby. So we can indirectly diagnose what's it called, Toxoplasma gondii, by doing a immunofluorescence assay for IgM antibody. Okay. Now we don't use IgG because IgG usually comes from the mother. Okay, so the IgG might be falsely elevated because of the mom. So that's why we look at the IgM. Okay, I hope that makes sense. So we don't look at tissue biopsies for babies. We look at the IgM. Now, for in terms of prevention, just be clean around cats, bro. Like it's not that deep. Obviously you want to cook meat to safe temperatures. Wash your fruits. Wash your food. Change the cat litter, in a proper way. Be careful of any soil that might have, cat poop in it. That's pretty much it. It's not that deep. Now moving on to the acanthamoeba species. Now the acanthamoeba species are, from the name, it's an amoeba, but these cause severe eye infections. Now, double circle on eye. Skin, central nervous system, all that stuff. Sorry for covering the slides. Now it spreads to the eyes through contact lenses, or cuts, or skin wounds, or it can even be inhaled in the lungs, okay? But mainly when we talk about acanthamoeba, we're scared of the eyes and contact lens use, okay? For example, we have acanthamoeba keratitis, which happens in healthy people, okay? So it can happen in healthy people, acanthamoeba keratitis, and it's an infection of the eye that can result in permanent visual impairment or blindness. It's very dangerous. You can see this patient's eye is, it's probably gone. I don't know what's going on here. So this is acanthamoeba keratitis, okay? Now, what's even more dangerous in immunocompromised patients is granulomatous amoebic encephalitis. It's a serious infection of the brain and spinal cord that typically occurs in people with compromised immune systems, and you can memorize the name pretty easily. It causes the formation of granulomas, and encephalitis is just an infection of the brain, or sorry, inflammation of the brain. So when you think of encephalitis, just know that this probably causes brain and spinal cord damage, and obviously it's in immunocompromised patients. Okay, so in terms of infectivity, trophozoites are the infective stage. So trophozoites here infect humans. Now, a bunch of other arrows show that, I think it shows that the cysts can enter humans in other ways. So why aren't cysts considered infective? It's because the cysts don't really cause the disease. The cysts turn into trophozoites in the body and then can maybe cause disease. But it's mainly trophozoites that are the infective stage. And the trophozoites and cysts can be diagnostic. So if you find them in corneal scrapings, tissue samples, that can be diagnostic of what's it called, acanthamoeba. Okay. So in terms of prevention, just clean your lenses. Just be careful with your eyes. It's very chill. This is when it comes to acanthamoeba, we're just scared of eye infections. So everything to do with eyes, just be careful with your lenses. I'm sure people who wear lenses know how tedious it is to make sure they're clean, and this is the reason why. Now, next, moving on, we have Naegleria fowleri. I think it's the brain-eating amoeba. Now, this amoeba can infect humans by entering the nose, okay? So it can, enter the nose as such during water-related activities. So usually when you take history from these patients, they'll tell you like in the past week or something, or the past 10 days, that I was swimming, okay? Now the amoeba travels to the brain and causes a severe infection called, what's it called? Where's my thing? Oh, there it is. Primary amoebic meningoencephalitis. Okay, let me just do this. And it's fatal. Okay, primary amoebic meningoencephalitis causes death in like 1 week. Okay, it's very well known to cause death very quickly. And this is the dangers of Naegleria fowleri. Now, in terms of the life cycle, there's not much you need to know, but the trophozoites are the infective stage here. Now, it also shows that the flagellated form causes infection, but it's mainly the trophozoites. And in Naegleria fowleri, we have the cyst stage, the trophozoite stage, and the flagellated stage. And the trophozoite stage is the infective stage here. Okay, and obviously swimming is a risk factor for this because you can inhale the trophozoites and get infected. Okay. Now, Naegleria fowleri, in terms of detection, you can either see the organisms in the CSF, you can detect the nucleic acids in the CSF by molecular assay. You can find antigens in the CSF by immunohistochemical staining. Because this is a kind of like a brain disease, you can always detect it in the CSF. Nowhere else, yeah. That's specifically where you detect it. Okay. Now, in terms of prevention, Just be careful. Niagara folaria is waiting for you. Don't swim in water with Niagara folaria, I guess. That's why we chlorinate pools. That's why we monitor a lot of waters. We make sure there's no Niagara folaria in it. Um, and we obviously put up warnings everywhere. That's kind of prevention wise. Next up, we have Trichomonas vaginalis. Here's the trophozoite dancing on a pole. This is kind of to signify that it is sexually transmitted and it resides in the female and male genital tracts. It's in the name, okay, vaginalis, okay? Now, this is special because it doesn't have a cyst form, okay? There's no cyst form for this organism, and the diagnosis is through microscopy for motile trophozoites, okay? Very simple. Now, in terms of prevention, just practice safe sex, use barrier methods for prevention. It's very simple, okay? And just if the drawing wasn't enough, here's what the trophozoites look like. And this is what, the life cycle of it looks like. It's a very, simple life cycle. The infective stage is the trophozoite. The diagnostic stage is also the trophozoite, and it's passed among, men and females through sexual intercourse. Very simple. Okay. So next up, we have the Babesia species. Babesiosis is a zoonotic infection that's transmitted by the Ixodes scapularis tick. Okay. Ixodes scapularis is a tick you'll see around a lot because it also passes a lot of diseases like I forgot what the disease was. It passes around a bacteria. I don't know if you guys took it, but Ixodes scapularis is a tick that also spreads babesia. Okay. Now humans are opportunistic hosts for babesia when bitten by the nymph. The nymph is the baby tick, or even the adult tick. So the baby and adult tick can pass on babesia to humans. Okay, and it's sometimes misidentified as plasmodium in malaria endemic areas. You're gonna see why because the life cycle of babesia is very similar to plasmodium. So when a tick bites a human, okay, what's it called? The parasite, enters the human. The sporozoites are introduced into the host, okay? Now these sporozoites can go in a cycle. They go to the red blood cells, become trophozoites, and also become merozoites, okay? So this is why it's very similar to malaria because, the sporozoites enter the blood, but there's no erythrocytic cycle. It goes directly to the red blood cells and becomes a ring-shaped trophozoite. And there's also this cross-shaped or whatever it's called, merozoites, okay? So the sporozoites, once again, are introduced into the host, like I said. And what's it called? We said that human-to-human transmission is also well recognized to occur through blood transfusion. So if you, had the trophozoites in your blood and you decided to give someone a blood transfusion without checking your blood, you transfer babesia to this patient, okay? Now, in terms of what's it called diagnosis, you can take a thick or thin blood smear stained with Giemsa, and you can see the Maltese cross. This is the merozoites that I was talking about. Okay, see the Maltese cross here. This is merozoites, and you can also see trophozoites. Now, what differentiates this from plasmodium is that you have intra and extra erythrocytic forms. Okay, it's very important to know this. Look how you can see trophozoites outside the red blood cells. You can also see them inside the red blood cells. That's very important to differentiate between babesia and plasmodium. And this is, like I said, they look very similar. So this is why babesia sometimes gets misdiagnosed as malaria. Now, in terms of prevention, at the individual level, it includes checking for nymphostage ticks at the end of each trip into a wooded area. So, I don't know, if you're going camping or going into wooded areas, just be careful of ticks and obviously cover up to make sure you don't get bitten by ticks, okay? And now we're finally going to move on to the helminths, okay? The metazoa. These are multicellular organisms. So first off, we have helminths, which are roundworms, okay? We have platyhelminths, which are flatworms. So we're going to talk first about the helminths, which are the roundworms. So let's get into Ascaris lumbricoides, okay? Ascaris lumbricoides, the transmission is very simple. Someone ingests an egg of Ascaris lumbricoides. Now, these eggs enter, the liver, go to the portal circulation, and they end up in the lungs. Now, when the larva ends up in the lungs, obviously it causes irritation on the lungs, so you cough it up. And when the patient coughs up the larva, it ends up getting swallowed, okay, going down to the gut. So the larva goes to the small intestines and become mature male and female worms, okay? And from the male and female worms, there will be eggs that'll be passed on into the feces, okay? So the male and female worms produce eggs in the small intestine, and those eggs go with your whatever, your food, your waste, and end up in the feces. So obviously, and you're gonna see for most of these helminths, the way we diagnose it instantly is through stool examination. I think actually a lot of these parasites are stool examination. So we want to look for the characteristic eggs and even sometimes the worm, which is kind of disgusting. But Ascaris has three kind of eggs. You have the unfertilized egg, looks like this. This is the fertilized egg, and then this is the egg that contains the larva. And this is going to be the infective one if it's ingested. Okay, so that's about it for diagnosis. Now Trichuris trichuria. This is what Trichuris trichuria kind of looks like. These are drawings once again, and the life cycle is very simple once again. It's kind of like Ascaris, where you ingest an egg, but doesn't go through all this lung stuff. The eggs just kind of hatch inside the small intestines instantly, and it becomes an adult in the cecum. Okay, I think I have that written down. So the larvae migrate to the cecum, penetrate the mucosa, and then mature into adults. Now, when they mature into adults, what do the adults do? They make eggs, and the eggs are then passed into the feces, which we can use to diagnose, the infection. So when we want to diagnose the infection, we look for characteristic eggs in the stool. And Trichuris trichuria is known for its barrel-shaped with polar plug kind of eggs. So you can see these are the polar plugs here. Very characteristic of Trichuris trichuria. And you'll see these in stool. Obviously, once again, if you want to confirm that the patient has a parasite, you can do an endoscopy and see adult worms attached to the mucosa through the camera. Very interesting stuff. Next up, Enterobius vermicularis. Okay, Enterobius vermicularis is a very interesting one. I can't wrap my head around it. It's kind of funny. Um, Basically, like any other organism, any of the other ones we talked about, yani. Once again, you ingest an egg. The larva hatches in the small intestine, and there are adult female and male worms in the cecum. Once again, what do? They make eggs. But something special about the females is that the females, kind of migrate to the perianal fold, okay? And they lay their eggs in the perianal fold. Okay, when that happens, you'll notice that kids start to scratch their perianal folds. Okay, very itchy. It's very itchy for them. Okay, so what's it called? The eggs are in the perianal fold, and when they hatch, they migrate back into the rectum and into the large intestine. So it's very interesting to me. And this is very cool because of all the itching. We'll talk about it later, but the patients or the children specifically spread the disease more. So the way we want to diagnose this is it's very easy. We can see the eggs on the perianal folds. We can take tape. This is called the Scotch tape preparation. Take the eggs from the perianal fold, and we can see, these kind of smooth eggs, very characteristic of, what's it called? I already forgot the name. Enterobius vermicularis. Okay. And obviously, um, the females, once again, or the adult worms do migrate to the perianal folds. So we could technically, what's it called? Visualize them or observe them in the perianal skin. We can see the adult worms laying their eggs there. Okay. Now in terms of the hookworm, there's two types of hookworms. We have Ancylostoma duodenale and Necator americanus, two different types of hookworms. Okay. Now the hookworm life cycle is very simple. The life cycle is initiated when a filiform larva penetrates the skin. Okay, penetrates intact skin of a human. When it penetrates the intact skin of a human, it goes to the small intestine, develops into adults, and once again, what do? They release eggs in the feces. All of these release their eggs in the feces, and it's very easy for you to observe them under microscopy. Characteristic eggs in stool. You can see that this one is very granular, I guess. I don't know. It looks like a cloud. Okay, and that's characteristic of what's it called? This one, the hookworms. So the hookworm acts also. So it's very picture based. Make sure you have all the pictures memorized. Hookworms look very much cloudy. And obviously you can observe the adult worms once again under endoscopy, okay? Now, in terms of prevention and control, health education, good hygiene practices, safe disposal of human waste, and safe agriculture practices. So like I said before, method with what's it called? Enterobius vermicularis. Sorry. The one with this one. When the kid scratches their perianal fold, they're more likely to, get the eggs on their hands, for example. And kids don't really wash their hands. They might put it in their mouth. They might put it in their food. infect other people. So it's very important to educate these people, give them good hygiene practices, and obviously treat these patients as soon as possible to avoid the spread. Now, last but not least, for the roundworms, I believe, we have Onchocerca volvulus. Okay. Onchocerca volvulus is very scary, actually. You can see a worm in this guy's eye. And these are skin nodules. Okay. And we'll talk about the life cycle right now. It's one of the few ones that has like a vector, which is the black fly or the simulium. Okay, simulium. Okay. Uh, and this black fly basically just takes a blood meal, and then the larva enters our skin. Okay. So when it enters our skin, it then ends up in the subcutaneous tissue. Okay. And in the subcutaneous tissue, it matures into adult worms. So the larva becomes an adult worm in our subcutaneous tissue. And this forms nodules. Okay. This is the nodules I talked about earlier. Now, these adults then make microfilariae. Okay. So the adults produce microfilariae. And these end up going to the skin, the eyes, other tissues, basically everywhere in the body. Okay. And then obviously, a black fly can come back, ingest the microfilariae by taking a bite of the skin or something, and the life cycle completes itself once more. So what's it called? So for example, we can do, to diagnose this, we can take a skin snip for microfilariae. We said that microfilariae go to the skin. We could also take a biopsy of the nodules. The nodules are very easy, okay? Like we said, the adult worms should be in the subcutaneous tissue. So we should be able to see the adult worms in the what's it called? The subcutaneous. Sorry, the adult worms in the subcutaneous tissue when we take a biopsy. Okay. Now, in terms of prevention and control, health education, vector control of the black fly, and then prompt identification and treatment should be very simple. So now we're gonna get into platyhelminths. Okay. Now platyhelminths are flatworms. Okay. These look flat, and I think there's only one important one that we're gonna go over. Now, platyhelminths can be divided into trematodes, which are flukes, and cestodes, which are tapeworms. Okay. Now, for cestodes specifically, they're segmented flatworms that have ribbon-like bodies. Okay. So this is the ribbon-like body. These are proglottids. Okay, we call these proglottids. Okay, and chains of them. All of this is called a strobilum. Important to know this. Okay, and the only one that we're going to talk about for this one is teniasis. Okay. So Tinea saginata, Tinea solium, Tinea asiatica, although we're not going to talk about this one much. All of these are forms of Tinea, and humans are the only definitive host, okay? So there's no other, animal that's a definitive host or anything. And humans mainly become infected by ingesting raw or undercooked cysticerci, in meat, okay? Either pig or what's it called? Cows, okay? We'll talk about it right now. So in the general life cycle, we have cysticerci. What's it called? Cysticerci are the cystic or the cysts in the meat of the cow and the the pig. Okay, so when a human ingests this meat, maybe undercooked or raw, he's gonna eat the cysts, which is kind of nasty, and the cysts are gonna end up in the intestines. Okay, and the cysts mature into adult tapeworms. Now, the adult tapeworms attach to the wall of the small intestines by their scolex. Now, the scolex is the head of the tapeworm. Okay, so the tapeworm attaches to the wall of the small intestine and just chills there, and it releases eggs or proglottids once again. So the proglottid, like we said, is the body of the worm. Okay, now the eggs are just the eggs, and both of these can be infective. Diagnostic. I'm sorry. Okay. Now the thing is, there's two different types that we want to talk about. We want to talk about saginata and solium. So Tinea saginata has its intermediate host as cattle. Okay, and the mode of transmission is through raw or undercooked beef, obviously. Now the diagnostic stage, like we said, is either the eggs, the proglottids, or the adult worms, and the infective stage is the larva, the cysticerci, the cysts, okay, in the animals. Now in solium, it's the opposite. It's the pigs, okay, larva in raw undercooked pork. That's the mode of transmission. But the interesting thing is that the infective stage of Tinea solium is larva and eggs. Okay, why is that? Let's see right now. So this is the life cycle we talked about right now. But you'll notice that there's this other life cycle, and this happens when you ingest the eggs. If you ingest the eggs of Tinea solium, okay, you're gonna get cysticercosis. And this is literally as if a pig ate the eggs. When a pig eats the eggs, he gets a cyst, right? If a human ate the egg as well, he'll also get a cyst in his muscle or his brain or his eyes. He'll just get some kind of cyst somewhere in his body. Now, this doesn't happen with saginatum. It only happens with solium. Okay, that's very important to know. So, how do we differentiate between the two? Well, first off, in the stool, both of them have characteristic eggs. Okay, but Taenia saginata, the adult worm, has a head that doesn't have a rostellum or a hooklet. This is the head of Taenia saginatum. I'm going to talk about what a rostellum and a hooklet are in a second. Now, there's also the proglottid, which is this. The proglottids have around 15 to 30 uterine branches in Taenia saginata. Okay, you can see all of these branches. These are all branches, if that makes sense to you guys. There's around 15 to 30 in Taenia saginata. On the other hand, Taenia solium, unlike saginatum, has a rostellum and a bunch of hooklets coming out like this. Okay, that differentiates them a bit. Other than that, Taenia solium proglottids have 7 to 12 uterine branches. So if we count this, I don't know how much it's going to come out to, but its point is it's way less than Taenia saginata. Okay, important to keep in mind. Now let's talk about Echinococcus granulosus. Okay, now Echinococcus granulosus has a definitive host being canines, which are dogs, and the intermediate host being herbivores or humans by accident. Okay, humans are either accidental or dead end hosts, so it comes to us by accident, and obviously the cycle can't continue after us, so we're dead end hosts, if that makes sense. Now the infective stage is a larva for the dog, the definitive host, but for what's called for humans, it's eggs. Okay, so ingestion of eggs of Echinococcus granulosus can cause this disease in humans. Now, the diagnostic stage, what's it called in humans, is the hydatid cyst. Okay, and we're going to talk about that in a second. So normally, a dog has an adult worm in the small intestine, and this adult worm releases eggs that are ingested by sheep or human. Okay, now it results in the same thing whether or not it's ingested by a sheep or a human. If it's ingested by a sheep or a human, okay, these eggs are then going to develop into large filled, what's it called? Hydatid cysts. These hydatid cysts can be in the liver, the lungs, the brain, and the bone. So imagine having a cyst, in your brain, lungs, or bone, or whatever. It could, be anywhere. Okay. And obviously, when it comes to sheep's, at least, the life cycle is completed when, this infected organ with the hydatid cyst gets eaten by the dog. Okay, the dog can eat it and uptake it again. Okay, so let's say like a liver containing hydatid cysts, for example. So what's interesting, or what you should know, is that the larva of Echinococcus granulosus causes unilocular hydatid cyst disease. Okay, so that means there's only one cyst being caused by this disease. But if we had more than one cyst, multilocular, it would be caused by Echinococcus multilocularis. Okay, tell me. Now, Echinococcus granulosus is composed of a scolex, okay, and only three proglottids. Okay, so it only has a head and then three, proglottids right after it, making it the smallest tapeworm out there. Okay. Now to test for it, it's very hard to test for Echinococcus granulosus because it causes a cyst in the body. But you can do an intradermal test, so you can inject someone with hydatid fluid. It's called the Cassoni test. If there's a reaction, they might be infected by what's it called, Echinococcus granulosus. You can also do microscopy of fluid aspirated from the cyst. So let's say you do an ultrasound of a patient and you see a cyst in their liver. And let's say you aspirate the what's it called, the fluid in the liver. Okay. You might be able to find, a high, what's it called, a hydatid brood capsule with prostoscolices. Okay, prostoscolices are just, the infective stage in dogs. These are basically the thing that infects a dog. So let's say, for example, a sheep, had a cyst. The cyst would have protoscolices, and when the dog eats the infected organ, the protoscolices are going to infect the dog. Okay, so in terms of prevention and control, we just want to cause, or sorry, for prevention and control, we just want to properly, thoroughly cook the meat, avoid contamination of grazing sites, don't feed random organs to dogs, and obviously, adequate treatment of patients. Okay, I hope that makes sense. Okay, so now we're going to continue on with what's it called? Oopsies. We're gonna go specifically talking about the trematodes from the platyhelminth, what's it called, group. Okay, so for trematodes, these are known as flukes, right? Now, trematodes, unlike the what's it called, the cestodes, okay, the tapeworms, they're non-segmented. So trematodes are non-segmented. They don't have the proglottids like the cestodes, right? So they're flat, fleshy, leaf-shaped worms. Okay, not just that, they're also hermaphroditic. Okay, so that means they act as both genders sometimes. So it can be a male and female in just one trematode. Okay, now the exception for this is schistosoma, which is the organism we're going to talk about. And for most trematodes, the eggs tend to have a lid, an operculum. Okay, but once again, schistosoma has a spine instead of a lid for its eggs. Okay, so we're going to talk about that in a second because the only organism we're going to talk about is schistosoma, anyways. Okay, so this is the spine I was talking about. Okay, and what's it called? Schistosoma isn't hermaphroditic. Okay, it has both male and females. But anyways, the definitive host for schistosoma is humans. Okay, the intermediate host is snails. Okay, snails is a very interesting one. The bullinus snail. Okay, we took this in the renal block. Okay, the snails are the intermediate host, and the habitat, depending on the species, is usually the mesenteric or cervical plexus. Okay, and the infective stage is something called the cercaria, which is basically just the larva. But just remember the word cercaria. And the diagnostic stage is eggs in the stool slash urine. Okay, so for most of these, once again, you're going to find eggs in the urine or the stool. Okay, urine is special in this case, and I'm going to talk about the specific species that you'll find in the urine. And the mode of transmission is through skin penetration. Okay, so in the life cycle, a cercariae, okay, this one, will infect someone swimming in fresh water by penetrating their skin. Okay, and then they become adult worms. Okay, so it migrates to the what's it called, the mesenteric plexus, and becomes an adult worm. Okay, so when it becomes an adult worm, the adult worm lays eggs. Okay, now these eggs can either appear in the stool or it can appear in the urine. Okay, the urine is a special one. Okay, so once these eggs pass on to the fresh water, what's it called? It becomes a what's it called, a mericidia. So this is a mericidia. The mericidia goes to the snail, infects the snail, and then the snail produces cercaria. And the cercaria is the infective stage that bites us and penetrates our skin and infects us. Okay, cercaria. Okay, mericidia is what infects the snail. Okay, and all of this happens in fresh water. Okay, I hope that makes sense. Now to specifically talk about each species, first off, Schistosoma mansoni, specifically found in the stool. This egg is found in the stool. Okay. Oh, I did a capital L there. Stool. Okay. Has a large lateral spine. Okay. You can see that it's lateral and it's pretty big, to be honest. Unlike Schistosoma japonicum with a small, small lateral spine, this one's also found in the stool. Okay. Schistosoma japonicum and Schistosoma mansoni both have lateral spines and they're both found in the stool. Okay. The difference is that Schistosoma mansoni has a large one. Japonicum has a small one. Okay. Now, hematobium, Schistosoma hematobium, this is the one that you find in the urine. Okay. You can find this egg in the urine, and it has a large terminal spine. Okay. This doesn't seem too large, but this is considered large. Khalas. It's the large terminal spine. It's the only one that isn't on the side laterally. This one's, once again, terminal. Okay. It's terminal spine. Now, in terms of prevention and control, proper disposal of human waste, eradication of the snail host, health education, swimming in, areas that, you know, don't have schistosoma, hopefully. Like, just be careful of that. That's prevention. It's very common sense, you know what I'm saying? So now let's switch gears. We're gonna be talking about fungi, okay? Now, fungi, Salaha, there's not much details for you guys to know. She kind of just threw this at you guys. The fungi lectures are always the worst, but hopefully I'll divide it into, something understandable. Hopefully you guys can get it. So the fungi are multinucleated and multicellular, okay? Now, for the most part, they are multicellular and multinucleated, but, fungi has different forms, one of them being yeast, and yeast is unicellular, okay? But for the most part, fungi is a multinucleate, multicellular organism, okay? Now, fungi, unlike us, they digest their food before they ingest it. So that's like if I, spit out my stomach acid, ate a sand, like dissolved the sand, which is my had, and then ingested it. Okay, and this is called being saprophytic. Okay, that's what's important about fungi. Now, it's important to know that the asexual form of fungi is an anamorph, and the sexual form is a telomorph. Okay, teleomorph is sexual, asexual is anamorph. Okay, and fungi are opportunistic pathogens for the most part. So when we say opportunistic, we mean that they quite literally take the opportunity when they can. So let's say you have a patient who's immunocompromised, and he comes to you and his tongue has fungi on it. That's because he's immunocompromised. For the most part, normal humans do a great job of getting rid of fungi from their body. For those who are less fortunate, immunocompromised, fungi can take the chance and infect the patient. Okay. So let's talk about the basic structure of fungi. Now, the fungal cell wall is primarily made of something called chitin. Okay. This is the cell wall we're talking about, chitin. Okay. On the cell wall, you can also find alpha beta glucan, which is a long polymer of D-glucose. You can also find mannans. Okay, composed of mannose-based polymers. These are all found in the cell wall, but it's primarily made out of chitin. Okay, I really guys want you guys to focus on chitin. Now, the cell membrane, on the other hand, contains ergosterol. Okay, that's the cell membrane, not the cell wall. Don't confuse it. Let me make like a divide here. So the fungal cell membrane contains ergosterol. And like we said earlier, fungi can exist either as mold or yeast. And we said yeast is what? Unicellular, صح? Okay, perfect. So this is just a picture. I talked about the ergosterol on the cell membrane. And I talked about the chitin, which is the main, structure thing of the cell wall. You also have, alpha beta glucan and your mannose proteins, okay? The mannan proteins. That's basically it for the fungi itself, yeah. Now, in terms of the, oops, did I do something? In terms of the different forms, okay, of what's it called? A fungi, you have first molds. Now, molds are literally just branching thread-like tubular filaments, okay? We call these hyphae, okay? To simplify, we call it hyphae. This is what a hyphae looks like. So you can see that it's branching and you have a bunch of, this is literally like what it could be like. I don't know. I'm just drawing. This is what a hyphae is. This is what mold is. And a mass of hyphae is called mycelium. Okay, so this is all of it is a mycelium. Specifically, this is hyphae though. Okay, so this is what a mold is. That's pretty much it. It's just tubular filaments, and yeah, this is what mold is. Now, mold can either be aseptate or septate. Now, aseptate hyphae is like this. You can see that it's multinucleated. Um, there's no borders between each cell. It's just all one cell. Okay. This is aseptate. It's hollow, multinucleate. It's also known as coenocytic. Okay. That's aseptate. Now, septate hyphae, you have clear divisions between each cell, very obvious divisions, and each one has its own nucleus, contains a nucleus, and this is called septate hyphae. Okay. Now let's move on to yeasts. Yeasts are unicellular. Okay. Usually round fungi. Wait, I feel like I missed something very important that I want to talk about. Okay, not yet. Okay. So yeasts are unicellular. They're usually round fungi. So you can see round fungi here. Uh, Trust me, this isn't some kind of bacteria. This is actually yeast. The shape looks a bit different from bacteria, but I think this is a gram stain, which I'll talk about later. So these yeasts are unicellular and they're round fungi. And some of these yeasts can actually, make something called pseudohyphae. Now the yeasts, the way they kind of divide is they butt off of each other. So you can see budding over here. This is budding of the yeast cell, budding of the yeast cell. When a yeast cell buds off, sometimes the budding is so long that it looks like a pseudo like a hyphae literally. And these are called pseudohyphae because it's a fake hyphae. It's not actually a hyphae. So these are just yeasts, unicellular organisms, or one cell. Okay, so this is what I wanted to talk about. Specifically morphologically, we said that fungi can either be mold or yeast. Okay, now there are dimorphic fungi that can either be mold or yeast depending on temperature. So there's a mnemonic that says, cold for mold and heat. Sorry, my handwriting is bad, but yeah, heat for what? Heat for yeast. I'm just writing fast. I want to, try and not spend as much time as possible. So yeasts happen at like 37 Celsius. Molds are at lower temperatures, like 22 to 25. So that's why we use the mnemonic cold for mold and heat for yeast. Okay, it makes a lot of sense. But I will tell you guys later about an organism that what's it called that flips this around. But for the most part, all of them follow this. Okay, there's another one that kind of just flips it. Okay, so then there's also something known as dematicious fungi, which are dark colored, usually from soil. And then you have hyaline fungi, which grow what's it called in a filamentous form, so hyphae. And the hyphae are colorless. Okay, you don't really see much of a color to them. Okay. Now, in terms of classification, she unfortunately does want you guys to know this, and it kind of sucks because it doesn't help you much as a physician. But we have first off Ascomycota, and just some things you need to memorize straight up. In Ascomycota, the sexual spores are in a sac called an ascus. Okay, and in these there's sparsely septate hyphae. So they are in hyphae. So Ascomycota, they do have hyphae. And it is septate, but the septa are kind of sparse, so like they're farther apart from each other, if that makes sense. But in Basidiomycota, it's septate, it's normal septate hyphae, okay? And the sexual spores they make are on Basidia, okay? So just memorize the word Basidia for Basidiomycota. It makes sense, hopefully, Annie. And for Zygomycota, they do sexual reproduction via gametes, okay? And these guys are aseptate hyphae, okay? So they don't have any septate, like we said, multinucleated hyphae. That's what zygomycota is. Now, deuteromycota, although it says in the slides that we don't use this classification anymore, they are fungi that have no known sexual stage, and this kind of includes most pathogenic fungi. Now let's go a bit deeper into each one. So in Ascomycota, the sexual spores are formed, like I said, in a sac-like structure known as an asci, right? And they also form asexual spores, and these are called kinidiophores. Okay, they're formed on kinidioforms. Okay, that's it for Ascomycota. For Basidiomycota, she just wants you to know that there's Cryptococcus neoformans and Cryptococcus gattii. These are important, what's it called, fungi in this classification that cause cryptococcosis. So in immunocompromised individuals, you can have like cryptococcosis in the lungs, the CNS. That's kind of all she wants you to know about Basidiomycota. And like I said about Basidiomycota, it has septate hyphae, and they form their spores on Basidium. Okay. Now Zygomycota, you can see the aseptate hyphae here. It's aseptate. Okay, there's no divisions between each cell, and they form sexual zygospores. Okay, so we said that they form their sexual their sexual spores are through reproduction of gametes. Okay, so this is why it's known as a zygospore. Okay. Now, in terms of mycosis, in terms of disease, now this is the actual important part of the lecture. We divide it based on clinically. We divide it based on where it actually causes disease. Okay, so first off, you have disease that's superficial, so superficial mycosis. You have cutaneous mycosis, you have subcutaneous mycosis, and you have systemic mycosis. Okay. Now, for superficial mycosis, it only involves the dead layer of the skin. Okay, so it's a cosmetic effect only. It doesn't harm the patient, aside from the cosmetic aspect, which is technically still harm because the patient looks, hideous or whatever they want to say. So this is why we have to treat this. Okay. Cutaneous mycosis. Now, this is actually kind of getting worse because now it's invading the epidermis and deeper levels of the hair, skin, and nails. Okay. Now, cutaneous mycosis is caused by something known as dermatophytes. Now, dermatophytes, we divide them into geophilic, zoophilic, and anthrophilic forms. Okay. So, geophilic, I want you guys to think of soil. That's for geophilic. For zoophilic, animals. For anthrophilic, I don't have space to write it. Humans, okay? My handwriting is ugly. I'm sorry. Humans, okay? That's anthrophilic. Now, what you want you guys to know is that geophilic and zoophilic forms cause acute meaning fast infections, while anthrophilic forms cause, slow, time-taking infections that, take a long time to, or stay for you with a long time. These are chronic infections, okay? So these are the different types of dermatophytes, and they cause cutaneous mycosis, okay? Next up, we have subcutaneous mycosis, usually through traumatic inoculation. I'll get into more details about that later. But these, invade the subcutaneous tissue, muscles, fascia. It goes way deeper than just the dermis, and these are very dangerous, okay? Systemic mycosis, even more dangerous than subcutaneous. These can often originate in the lungs, but can literally spread anywhere. And they're often caused by dimorphic fungi in the immunocompromised. Okay, so once again, immunocompromised again and again. If you think of immunocompromised, always think of fungi, or the opposite. If you think of fungi, always think of immunocompromised patient. Um, and yeah, and there's a bit of others. Now, these kind of still fall under systemic. So you have opportunistic mycosis. These only occur in patients who are immunocompromised. Okay, so for example, thrush. Oral thrush is common in people who are immunocompromised. And mycotoxins, some fungi produce toxins that cause disease in patients. Okay, I hope that makes sense. And specifically, if we want to kind of visualize, these fungi under microscope or whatever, there are some preparations we can make. اللي هو potassium hydroxide, so KOH. It's a solution we use to visualize dermatophytes. Okay. We can also use a gram stain. We saw it earlier. Trust me, this is not bacteria. This is actually Candida. We can gram stain Candida. Okay. And last but not least, you have the India ink mounts. Okay. For Cryptococcus, the Cryptococcus species has this very kind of halo appearance, very typical of it. And you can visualize it because of the India ink mount. Okay. Now, a bit more about mycotoxicosis. Now, mycotoxicosis, when we diagnose mycotoxicosis, like being infected by a fungal toxin, it's usually based on epidemiological clues. Okay. So someone, ate contaminated food, or someone is known to work in a building that has a lot of mold. Okay. But, generally, mycotoxicosis is very hard to, diagnose, and it's challenging because of the varied symptoms and, the very toxins that are out there. Okay. So you mentioned this in the slides. I just put it in for completion's sake. Now, when we want to, what's it called, culture fungi, the traditional mycological medium is Sabouraud's dextrose agar, SDA. Okay, it contains glucose and modified peptones, and we use this to, what's it called? Culture fungi. Okay, this is a very important medium. Tamam? Now, let's get back into the different diseases that she wants you guys to know. First off, we have superficial mycosis. Now, superficial mycosis, like we said, infects the dead layer of skin. And the one she wants you guys to know is pityriasis or Tinea versicolor. Okay, pityriasis versicolor. It's caused by the Malassezia species. Okay, these are lipophilic yeasts. And you can confirm this disease by taking a skin scraping, treating with potassium hydroxide, and then staining with calcofluor white or lactophenol cotton blue. Okay, and I want you guys to memorize this just in case. I don't know. She did have it emphasized in the slides. So this is the stain you use to, visualize pityriasis versicolor. And under the microscope, you have a spaghetti and meatball appearance because you can see unbranched hyphae and spherical cells that are seen under the microscope. And clinically, if you use something called the Wood's lamp, you can see fluorescence of the actual lesion. Okay, so under lactophenol cotton blue stain, you can see, the balls. You can see the hyphae. You can basically see what I mean by spaghetti and meatball appearance, right? It looks very much like spaghetti and meatballs, kind of typical of Malassezia furfur, or pityriasis versicolor. Okay. Now, that's it for superficial. Now we're gonna move on to cutaneous mycosis. Okay. Now for cutaneous mycosis, we have a ton to go through. I think 3. So first off, we have Trichophyton tonsurans. Okay. Now Trichophyton tonsurans infects the hair, the skin, or the nails. Now, the way I remember this is the word tri. It starts off with tri. So tri is just the same as 3. So it infects hair, skin, nails. It infects 3 things. Okay. The others only infect 2. We'll see that later. So on culture, it's a powdery to velvety culture surface. Maybe you can see that, maybe not. It looks like this. But when we reverse it, we can see a reddish brown appearance. Okay. So that's for Trichophyton tonsurans. Now, when we actually do microscopy of Trichophyton tonsurans, we see cylindrical, smooth wall macroconidia. Okay, these are the spores with grape-like clusters of micronidia. Okay, so the micronidia are these, right? These are the small grape-like clusters of them. These big guys are the macroconidia. Okay, now the thing that's diagnostic for Trichophyton is that once again you can see grape-like clusters of the micronidia, and the macroconidia are also there. But it's mainly the grape-like cluster. So if you see clustering of these tiny microconidia, it's most likely Trichophyton. Now for microsporum, It infects only the hair and the skin, okay? And it has a white cotton surface on the culture surface. So the trichophyton had like a velvety kind of white appearance. This one's more cottony, okay? And on reverse, instead of reddish brown, you see a yellow surface, okay? Yellow color on reverse. Now, when we do a microscopy of this, we'll see spindle-shaped macroconidia and with ecunulate walls. So what does ecunulate mean? Echinodate basically means spiky, okay? So I don't know if I can draw properly or not. We'll try and draw. So this is kind of the macronidia. If you actually zoomed in, you'd see spikes, okay? I genuinely can't zoom in for the life of me, but you can kind of see the tiny... I zoomed in earlier. I swear they're there. There's spiky kind of things on the walls of the macroconidia. And of course, you can see, I didn't mention this on text, but there's micronidia everywhere, okay? Very small spores everywhere. Okay, this will help us differentiate from the next disease, which is epidermal phyton flocosum, okay, which infects only the skin and the nails. So the other one had the skin and the hair, right? Microsporum, skin and hair. Trichophyton, all three. Epidermal phyton flocosum, skin and nails, okay? It helps to, the way I correlated this is that like epidermis, epidermal phyton, epidermis, uh, skin and nails is close enough. That's kind of how I kind of correlated it. You guys can correlate it however you like. On culture surface, you're going to see an olive green color, and the yellow color is on reverse, okay? Just like microsporum, but the front is going to be olive green instead, okay? So what differentiates this from trichophyton and epidermal phyton is that it produces only macroconidia, okay? So macroconidia, they look very silly. I don't know. But when you see this, you should instantly know it's what's it called? You should instantly know that it's epidermal phyton because you don't see any microconidia anywhere. Unlike microsporum, where you can see obvious microconidia everywhere, it's not as common in what's it called? Epidermal phyton. Okay. Next, moving on to subcutaneous mycosis. Like I said, these ones are, they enter the tissue and skin by traumatic inoculation. Okay. Subcutaneous mycosis can enter the skin traumatically. Let's say like a metal rod just entered your skin, something like that. And these are very dangerous. The disease she wants you to know is Sporothrix schenki. It lives on vegetation and it's thermally dimorphic like most fungi. In the environment, you see filamentous hyphae, which is mold. And then in the host tissue, it's yeast. Okay. There's not much to really know about this one. I don't think, she gave you guys much to work with. But when we do microscopy of this, you'll see that, the hyphae are septate, okay? So that's first off. You can see, the septas right here. They're very visual, at least on my screen anyways. I hope you guys can see them along with me. And then the conidia are sometimes, referred to as resembling a flower. So you can see this one, for example. Sorry, I covered it a bit. But you can see, like, yeah, like, it kind of does look like a flower in and of itself somehow. If you use your imagination, this is a flower to some degree. And this is, characteristic of Sporothrix schenki. Now we're going to move on to the systemic mycosis, sorry. First off, we have histoplasma capsulatum, okay? And it's the most prevalent pulmonary fungal infection in humans, okay? Very interesting, right? It's usually associated with avian, so birds and stuff, habitats like that. And the way we diagnose this is by finding ovoid cells inside of macrophages, sorry. So inside of macrophages, we can see small ovoid cells, and this is, diagnostic for histoplasma capsulatum, okay? Next up, we have Candida species. There's a bunch of different species of Candida. The most important one is albicans, Candida albicans. These are opportunistic dimorphic fungi. And these are the ones that I told you that flip the script a bit. So listen to this. So first off, they are yeasts, okay? They're oval budding yeasts, and they form pseudohyphae. So when Candida starts to bud off from another cell, it kind of sometimes pulls it with it. It's like a cheese pull. It forms a pseudohyphae along with it, okay? Now, what I meant by it switches things around is that when we put it in a lab environment and raise the temperature, it starts to become a mold, okay? Remember how earlier we said cold is for mold and heat is for yeast? Candida albicans flips it around. So in this case, cold is for yeast and mold is for heat. So when we raise the temperature at higher temperatures, we're going to get something called a germ tube. And this is, you know, the production of true hyphae. So it's becoming mold. Okay, we're gonna see that right now. So first off, this is the pseudo hyphae. So you know, it's causing budding cells. These are budding cells. They're forming pseudo hyphae, but they aren't actually hyphae. And when we raise the temperature, we have germ tube formation. This is a germ tube, and it's a true hyphae at higher temperatures. Okay. Next up, we have the Aspergillus species. It's a mold that produces small conidia that can easily be aerosolized, so we can inhale it easily. Now, Aspergillus species is special because it's not dimorphic. Aspergillus species is not dimorphic. And depending on where the disease is, like if it's a pulmonary disease, we can take a sputum sample from the patient. We can do a something called a bronchial washing. We can take a tracheal aspirate. We can take a lot of stuff. Basically, anything related to the airways, we can take a sample from that to detect Aspergillus. We can also, what's it called, do tissue biopsies from those with disseminated disease, all that kind of stuff. Okay. And as you can see, you know, Aspergillus can infect the lungs once it's inhaled, all that kind of stuff. So when we do, what's it called? When we do microscopy, we see septate hyphae that branch from each other at a 45-degree angle. So I don't know if it's visible here, but say this is a branching point. You could potentially say it's at a 45-degree angle, or this one branching off like this. If I was to put it in a way, it would look something like this. So they'd be branching off from each other like this at a 45-degree angle, somewhat something like that. Okay. And this differentiates it from another kind of fungi that you guys might take later. Not in this block, but there's another fungi that, you know, emerges at a 90-degree angle. Okay. But yeah, what's it called? This one, Aspergillus, goes at a 45-degree angle. Okay. Now, another thing about Aspergillus fumigatus is that, or just Aspergillus species in general, is that the conidia form radiating chains. Okay. So what do I mean by that? So look over here at this hyphae. You have what's it called? Conidia right here, right? Now the conidia start moving outwards in a radiating chain, and you can see a bunch of conidia radiating outwards, like such. Okay, this is what we mean by a radiating chain. Radiating chain. So it's very characteristic for Aspergillus. If you see something like this, know that it's Aspergillus instantly because of these radiating conidia, radiating chains of conidia. Okay, and that's pretty much it. Actually, surprisingly, thank you guys so much for your time. I genuinely hope I was able to give like a super high yield review of everything. I do apologize. I am a bit sick. I've been sick for the past week. I don't know if you guys can tell from my voice, but I hope I was still able to deliver regardless. But if I wasn't able to, this right here is a quiz. Okay, I put around 5 questions in this quiz. Um, you know, just to quickly test your knowledge. It's also leads to the feedback form. So once you finish the quiz, I'd love to get your guys' feedback on how I did. Um, I do want to know ways I can improve, stuff like that, especially because I am sick this time around. So I don't know if I was able to give a proper review of things, but hey, hopefully I was. So make sure to scan this code for, you know, the quiz. Um, see how you do, and what's it called? Make sure to give me some feedback, and thank you guys so much. That'll be it from me. If you guys have any questions, feel free to message me. And best of luck on your POD final, guys. Y'all got this, inshallah. Yalla. Bye bye. Let me stop the screen now.